I'd like to welcome everybody today. Aloha. My name is Lisa Canali, and I'm the program coordinator for the University of Hawaii at Hilo, Tropical Conservation Biology and Environmental Science Professional Internship Track. This is a graduate program. And one of the things that is a requirement for all the graduate students is to give a public presentation. And so today, this afternoon, we have the great fortune of being able to hear Wilson L. Malone's experience working in Alaska. And I'm very excited about this presentation that he's about to give you. And to kick things off, we're going to have his supervisor at the Alaska Department of Fish and Game, Jeff Perschbacher, introduce him. So Jeff, let me hand this off to you.
Great, thanks, Lisa. My name is Jeff Perschbacher. I work for the Fish and Game Department up here in Alaska. I met Wilson in 2017 when we hired him on a research project assessing king salmon returns on the Kenai River. He brought with him a lot of great experience. He worked as partially as a commercial fisherman down in Southeast Alaska. He was getting his undergraduate degree in biology and had just a general knowledge and interest in the outdoors. King salmon, sockeye salmon, all management of salmon up here it can be very complicated. People are very passionate about it. It affects people's livelihoods. It can be very divisive, and it's rewarding to see our technicians like Wilson who aren't dissuaded by this and want to pursue a master's degree to try to be part of the solution. And with that, take it away, Wilson.
All right, thank you, Jeff. Hello, everyone. Thank you for tuning in. My name is Wilson Malone. I'm here to talk about the sustainable management of Alaska's Kenai River Chinook salmon and its economic, cultural, and scientific contexts. So I would like to first thank my mentor, Robert Begich, for assisting me with the scientific and technical aspects of my internship project earlier in the year. And I would also like to thank my supervisor, Jeff, for providing stellar instruction on performing my job as a fisheries technician and providing help with editing my report. I would also like to thank Lisa Canali, my internship coordinator, for providing support in my academic pursuits and internship pursuits and for making this all possible. Now for a little bit of my background. I grew up in Alaska, and as such, I have a long relationship with salmon and the outdoors. Part of my early fishing experience was with Southeast Alaska's subsistence salmon fishery. By my teenage years, I was familiar with deploying drift nets, catching salmon, and contending with hungry seals who stalk the nets for a quick snack. I moved to the Kenai Peninsula years later, which, like my earlier home, has a significant salmon fishery. The Kenai Peninsula is an outcropping of south central Alaska that is bordered on the west by Cook Inlet and on the east by the Gulf of Alaska. The Kenai River runs from east to west and is centrally located on the peninsula. Let's see. Right here. Okay. So the Kenai River runs from about this area through here and exits there. So centrally located. The Kenai Peninsula, or the Kenai River, supports 4 anadromous species of salmon: sockeye, Chinook, also known as kings, coho, and pink salmon. Just looking at the pictures, you probably can't tell them apart, but don't worry. We have diagnostic methods of identifying them. Anadromous means that they migrate from saltwater to freshwater to spawn. They're semiparous, which means they die after breeding. Sport salmon fishing in Cook Inlet centers around 4 species of commonly caught salmon: sockeye, Chinook, coho, with pink salmon being of lesser demand. Sport fishers in the rivers will target trout as well. Chinook salmon are valued for their size and status as a trophy fish. Sockeye are an economically important fish for the commercial fisheries and are the most commonly caught. Salmon fishing has a long history in the Cook Inlet region. People of the Dena'ina ethno-linguistic group pioneered intensive salmon harvesting, which enabled the transition from a nomadic hunting way of life to a more sedentary fishing-oriented way of life. Numerous innovations, such as the construction of weirs and slow-moving streams and birch bark and moss-lined salmon storage pits, followed this salmon-intensive lifestyle. Today, multiple tribes are located in communities around Cook Inlet and still maintain a connection to a way of life grounded in the region's natural resources and fisheries. Up until Alaska's statehood in 1959, salmon fisheries were managed under federal rules, which allowed fish traps. Fish traps are a method of catching salmon that is highly efficient, but detrimentally so, as it takes advantage of high concentrations of fish as they migrate close to shore or in rivers. They consist of a maze of wire and wood, which guides salmon to a holding pen where they are harvested. This method encouraged excessive catch and tended to favor a smaller number of user groups as well as influential salmon canneries. A large part of the impetus for Alaska's statehood was preventing the concentration of fish resources into the hands of a few influential companies. The state's constitution outlines the principle that heavily utilized natural resources were to be managed under the principle of sustained yield, and that no single user group would be given preference over the other. Sustained yield is a mathematical concept within population biology, and I'll be covering that a little later. The Department of Fish and Game was given broad authority to conduct fisheries research in the state and to set escapement goals, while the allocation decisions were handed over to the Board of Fisheries, which consists of members appointed by the governor and representative of the fisheries in the region. Fisheries are vital to the economy of the region. Commercial fisheries support small local businesses that catch fish commercially. They're often family-owned and involve multiple generations of families. Sport fishing brings tourists into the region and provides a business ecosystem for river fishing guides and local retailers. There are multiple user groups and advocacy organizations that represent the interests of their own type of fishing, often in an adversarial manner. Commercial salmon fishing at Cook Inlet can be done by set net or by drift net. Set nets are tethered to the shore of the inlet, while drift nets are allowed to float into drift. Commercial fisheries don't target a specific salmon species, although sockeye make up a majority portion of the catch. Sport fishers in Cook Inlet and the Kenai River primarily use rod and reel to catch the 5 species of salmon as well as trout in the rivers. The popular personal use fishery is performed by permit only and is designed to give Alaskans a chance to have priority over out-of-state users. In this picture, we're looking at users of the personal use fishery in the Kasilof River near where I lived. Because they all target many of the same species of fish and often the very same populations, there is a contention between user groups. Each group has at one point blamed the other for having an outsized portion of the catch. But this contentiousness demonstrates the importance of the fisheries to the local area, people, their livelihoods, and the economy. Now that brings us to the actual job of a fisheries technician. Here's a picture of me at the sampling site wearing my official Fish and Game issued hat and life jacket. We work in an area of low-lying grassland and tidal mudflats where it grows the occasional scrubby spruce or willow tree. Our sampling area is at river mile 8.6, which is measured from the mouth of the river. The area supports a small herd of caribou, and bears will sometimes visit the water's edge when no one's around. Eagles are a common sight, and we have to contend with opportunistic seals that scavenge fish out of our nets for us. The proximity to wildlife is one of the most unique aspects of the job, and it's the one that I appreciate the most. My project focused mainly on the in-river testnet fishery. The purpose of this is to provide data on catch per unit effort, also known as CPUE, and to provide proportion by age for Chinook salmon. Genetic samples that we gather inform about the subpopulations within the river. Data provides a layer of data on top of sonar count information, which leaves out important data points that can only be gathered by sampling the fish in person. This is our research boat. We use a flat bottom aluminum boat with a jet unit for operating in shallow water. Here you can see my coworkers constructing a remote shelter for use during the sampling season. Here's a short clip of what it's like to drive on the river. So as you can see by the layers of coats that we have to use, it's very cold. So next is shown is the process of casting a net from the boat. And next we'll show a brief segment of how to sample a sockeye salmon. First, they're measured, then a clipping of their axillary process is taken, usually with a set of clippers like shown. The axillary process is a modified scale that appears behind the pelvic fins. We generally keep this tissue sample in ethanol for later sampling. And when we're done, we release them. Sampling a Chinook is different. However, they are not removed from the water for a few reasons. Firstly, to avoid undue stress, and because they're often large and belligerent. We measure them and determine sex based on diagnostic characteristics, namely the ratio of eye to snout length to their total length, as well as the presence or absence of an ovipositor. We use a cradle designed to keep them underwater during the sampling process. The DNA sample is from their dorsal fin, and 3 scales are taken and placed on an adhesive card. And here's a short video of that. Here we're taking the scales. And here's a video of a large male being released. So fish age is provided by scale samples, which have rings that are interpreted. We collect 3 scales from each Chinook that we catch. The scales themselves are pressed onto an adhesive card. The scale patterns of 1 year of growth are called annuli, and we rely on the width between circuli, which is an individual ridge, to inform about the salmon's growth rate and life history. This will be interpreted into an age to give seasonal growth and ocean versus freshwater growth. The scales are later hydraulically pressed onto acetate cards and read with microfiche. Here we see a transferred impression of Chinook scales at the end of our sampling season. Our data entry is facilitated by weather-resistant field computers. On the right is an example of a Wattman genetic card and an adhesive scale card. We write the sex of the fish along with its length in millimeters to keep track of them. Sonar stations form a vital part of the management strategy of the river. There are multiple sonar stations in the Kenai River that capture all of the populations and species that migrate. However, the Chinook sonar is located at river mile 13.7, a few miles upriver of our in-river net sampling site. Sonar counts of fish capture a broad snapshot of every species and subpopulation that migrates. This information is useful for a broader picture of the run, but it does not tell you what species appear in the image. Some estimations are made, such as that all fish above 75 centimeters are assumed to be Chinook salmon, and below this length, the proportion by species are calculated from data obtained through other projects to inform about those. The sonar stations are adaptive resolution imaging sonars, or ARIS, and are an upgrade from the older split beam type sonars. One of the tools that Department of Fish and Game uses to gauge the social element of fishing is the creel survey. Creel is an archaic term for a fishing basket. Therefore, to do a creel survey is to count how many fish everyone has caught. What we do in this position is perform a boat count that travels between two fixed locations on the river, in this case, the bridge and the mouth of a specific creek, which we'll show later. We do a count of the number of boats and the number of lines in the water between these 2 points on the lower Kenai. When the survey is done, creel technicians then wait at one of several commonly used boat launches and interview anglers as they come in for the day. We take measurements, scales, and genetic samples from Chinook, the same as the netting crew. We also ask where the group was fishing, for how long. All of this helps the Fish and Game to gauge how much the fishery is being exploited, and it serves as an important point of contact between Fish and Game and anglers. So here's an overview of our sampling area. So we start to the left here, we have the Warren Ames Bridge, and then we travel and do a survey throughout all of this region through here up until Slycock Creek over here, just downstream of this bridge. Technicians are often called on to participate in other projects besides their primary focus. Besides Chinook salmon sampling, we were tasked with deploying radio trackers and Sockeye salmon. The radio tracking project sought to find the migration times of the various populations of Sockeye. This is going to be determined with genetic samples to calculate the proportion of Sockeye from each population, which will be coupled with run timing to better predict when a wave of fish will reach their upper tributaries. During 2019, we were met with an unusual wave of pink salmon, which normally would only migrate into the Kenai during even-numbered years. Fish and Game biologists took notice and tasked us with collecting otoliths, or ear bones, from the ones we caught to help determine where the fish came from. So how that would work is that they would examine the ear bones. They would find the pattern of rings imprinted on it through thermal cycling in the hatcheries where they were reared. That could help determine if they're wild stock or a hatchery, and if hatchery, which one they came from. I was also able to participate in a rainbow trout survey that sought to find the range of lengths of the upper river population, as well as the catch per unit effort for the species. Department of Fish and Game for this project uses their own drift boats, pictured here, which are launched at an upper Kenai River boat launch and then allowed to drift downriver. A number of technicians are tasked with fishing from either shore or the boat at various predetermined locations. I was also able to be briefly rotated onto the east side set net crew, which is the commercial set net fishery equivalent to our in-river netting project. They also focus on sampling Chinook salmon, but ones that are commercially caught instead. Genetic testing in the fishery helps inform of the ways that a species separates into different populations. Chinook salmon have two distinct runs that are genetically separate, an early run and a late run. They are geographically separated because the early run spawns in the tributaries and the late run spawns in the main stem. This graph shows a distinct bump in between the early and late run Chinook salmon based on the timing of the run. Salmon are extraordinarily specific in the location they choose to breed. often returning to the exact same stream they spawned from. Sonar data cannot differentiate among populations because of their location-specific breeding behavior. Salmon subpopulations within the species are genetically isolated enough to identify with microsatellite or S&P loci. Both Sockeye salmon and Chinook salmon are of interest genetically in the Kenai River. Recent efforts to identify the proportion of each run by subpopulation seeks to better understand the run timing of the migration. This will enable faster response times in management decisions. So when I'm talking about tributaries, what we see here is the main stem of the Kenai River, followed by a number of tributaries that feed into it in the watershed. So to accomplish their goals, Fish and Game operates the Gene Conservation Laboratory in Anchorage. Here they engage in sequencing and comparing the genetic samples that we and other teams collect over the course of the seasons. They use the latest in gene sequencing technology to give very fast turnaround times that enable fast management decisions about how to restrict or unrestrict fishing for specific populations and specific tributaries. Salmon spend some amount of time in freshwater immediately after hatching, and then later migrate out to sea. They return to spawn at varying ages, so it is helpful to think of a population of salmon in terms of its brood year, which is simply that year that a generation of salmon spawned. Proportion by age is one metric that Fish and Game uses to gauge the health of the population. By looking at long-term trends for the Kenai, we can tell the proportion of 5-year Chinook salmon has been going up, while the proportion of 6-year Chinook have been decreasing. Because fish age is highly correlated to their size, this represents decreasing average size across the board of the salmon species. This is a trend that has been seen across Alaska River systems in general, not just the Kenai. Several reasons for this have been proposed, including fishing-induced evolution, predation by marine mammals, climate change. No definitive answers have been presented yet. However, it is likely a combination of factors. In regards to warmer temperatures, I have directly observed fish becoming lethargic when river water temperatures are above normal, which is a process that can affect spawning behavior. If anyone here is familiar with population biology, this type of graph should look familiar. This shows one calculated relationship of recruits to spawners for the late run Chinook salmon, measuring 75 centimeters or above. This model is based on the Ricker stock recruit relationship, which models intraspecies spawning competition. This is a median range of possible spawner recruit relationships for this population, and there are upper and lower ends of the range that can be graphed separately and taken into account. So this vertical dashed line represents the point of maximum sustained yield, what we were talking about earlier. The solid line represents the point where recruitment is exactly equal to number of spawners. And this arched dashed line represents the stock recruit relationship. Current management goals call for an escapement of 13,500 to 27,000 Chinook salmon that are 75 centimeters or longer. And this calculation is derived from historical data and models that model stock recruit relationships. When we look at productivity in this population, specifically recruits gained per spawner for the late run Chinook, we see a drop off in the ratio of recruits to spawners over time, with a steep drop off in 2003, suggesting that there is something impeding the survival of salmon from between the time they are eggs to the time they return to spawn. The negative points here on this graph or where the productivity falls below replacement levels, indicating a shrinking population. And that's reflected here in this graph that also shows the same trend of a steep drop off in population. One study that looks into this population has correlated higher precipitation during spawning, increased stream temperatures, and oceanic temperature conditions to lower productivity in Chinook salmon. And I have a reference to that in the lower right corner. So to see how this lower productivity in recent times would play out in terms of the fishery, I constructed a simplistic simulation that looked at the stock-recruit relationship over 20-year periods. It was my goal to see how changing values of productivity and harvest rate could affect the run and escapement size. I added realistic variations to the values of alpha and the harvest rate based on historical data for the late run Chinook salmon over 75 centimeters. I ran this 20-year period simulation 1,000 times to give a large sample of possible future salmon runs based on this model. What I have here is a ratio of recruits to spawners for each of those simulations that's fitted to a regression line, a logarithmic regression line. So what I found was that there was a median of 2 lower escapement goal failures for 20-year periods starting in 2019. This held true even with a 30% reduction in harvest rate across all user groups. A mean escapement failure rate was somewhere around 6.5. And for the full set of 1,000 simulations, each of these graphs represents a different set of 1,000 simulations. These error bars represent the lines, the upper and lower bounds of the failure rates for those simulations. So the fact that, in fact, a lower harvest rate had no significant effect on the number of escapement goal failures. So the median values had 6 and 6.5 failures over the 20-year period. So my simulation had the limitation that it was treating all the runs as if they were the same age group, whereas in reality, a run is a mixed age population, as I explained earlier with the graph that showed the age proportions over time. So you can expect this would have some effect on the results. It may be of interest to note that the 2019 year run did not reach the lower escapement goal. Now, the reasons from this could be, as we were discussing earlier, and anything in between the time they are spawned to the time that they have to return. So what we're looking at is perhaps problems with their habitat or decreased survival in the oceanic phase of their lives. It will require looking at the entire life cycle in detail to figure out what is the point of concern here. Declining salmon numbers are of concern for riparian ecosystem contributions. Ocean-derived nutrients are a vital input for Alaska's riparian ecosystems. Salmon carcasses provide nutrients to bears, eagles, corvid birds, aquatic invertebrates, algae, other fish, and all of the plants surrounding the river. Oceanic derived nutrients find their way into the riparian ecosystems through salmon, and anything that causes a decline in salmon populations threatens to upset that input. Economic and cultural consequences will be felt as well in the face of a decline. Commercial fishing businesses and families in the region often make a tradition out of fishing. Native tribes in the region still rely on salmon for subsistence and cultural practices. With fewer salmon, their entire communities would suffer. Businesses in the towns surrounding the rivers would be hurt by a lack of interest in fishing if economically important species are returning in lower numbers. Even with the current declines, pain is being felt in the guide businesses with lower demand for Chinook fishing guides. Maintaining the fishery is important for the social and economic fabric of the region. Research and management of the type that Department of Fish and Game conducts is and will continue to be vital for maintaining salmon as a cultural and economic resource. Protection of riparian ecosystems and spawning grounds will also be vital to maintaining the healthy ecosystems that are built around salmon and their yearly input of energy and nutrients. Fishery of the Kenai River is being maintained well according to the tried and true fish population management techniques and the latest in genetic research. The Chinook salmon in particular have a challenge ahead as the run size and body size appears to be declining slowly. As always, riparian ecosystems protections are vital for the maintenance of salmon populations. Furthermore, oceanic research into the Chinook salmon as well as other salmon would shed light on the challenges of the life of salmon in the ocean. It could provide the answers for conservation actions that help preserve them and other salmon into the future. And that concludes my presentation, and thank you all for taking time out of your day to attend. Any questions?
Thank you, Wilson. If you have a question, please feel free just to unmute yourself and ask the question.
Hi, Wilson.
Hello?
Hi, great job on your presentation.
Thank you.
Yeah, I was just wondering, what was your favorite part of this experience, internship, master's program that you experienced?
Well, what I liked about this job specifically was working outdoors, working in an environment where I could, really be out in nature and, be in contact with fish and animals every day. And that was amazing. I really enjoyed the master's degree experience in terms of the opportunities for growing knowledge that it gave me. There were classes that I took, things that I learned that in my master's degree program that I never would have learned at the undergraduate level. And I found that extraordinarily valuable.
Thank you.
So Wilson, I had a question about one of the graphs. It's the one with the fish size. Do you mind bringing that back up?
Yes, this one. Or
Yeah, that's the one. So I just wanted you to clarify, is that data just from where you did your research?
this is, however, other studies and papers that I have read that address this exact issue have found these same trends across all Alaska river systems for the Chinook salmon. So it is a widespread population-wide problem.
Okay, and then the data, it represents the 3 different methods of data collecting, the Inlet, the Sonar, and the seine, or is it just the in-river?
Yes, this is all methods of collection
Okay.
between the 1980s and the present.
Okay. Great. So I guess then my follow-up question is, if you were to give the Alaska Fish and Wildlife Department, a suggestion of where to do their study next to kind of dig more into the population decline, where do you think they would have the most bang for their buck?
Well, I would say the ocean in particular. That is probably where a lot of the problem is happening. Something about their life cycle in the ocean is creating a kind of bottleneck for the populations. Other places I might look are river environments, temperatures, and rainfall patterns. One study suggested that too much water flow and too much rainfall at a certain time in the spawning cycle can be detrimental. So changes to rainfall patterns can also potentially affect how the productivity of the Chinook salmon.
All right. Thank you. I'd like to ask a question of Wilson and or of the presentation. Great. Then I guess we will stop the recording. Want to say thank you very much, Wilson, for your presentation today. Appreciate it. It will be on YouTube, and but not soon, but by January. And so thank you very much, and thank you all for coming today. Appreciate you spending the time with us. Right.
Thank you all.